Yeah, good morning, one and all.
You can use mine. You can use it.
Yes, sir, it's working.
Video also.
Yes, sir.
Yeah.
So I submit my humble pranams to His Holiness and blessings of Sri Jagadguru Deshikendra Mahaswamiji. A very good morning to one and all present here. So on this beautiful morning, I am pleased to welcome each and every one of you to a live webinar on state-of-the-art molecular modeling tools, BIOVIA drug discovery suite. A grateful thanks to Pro-Chancellor Dr. Suresh sir and our beloved Principal Dr. SP Dhanbal sir for giving this opportunity. And I truly appreciate the encouragement and efforts taken by my HOD Vice Principal, Dr. Mohammad Afzal Azam sir, and my fellow colleagues for conducting this webinar. So without any delay, let's enter the session. First of all, I welcome our today's speaker, Ms. Divya Shanmugrajan, Altem Technologies. Welcome, madam. And I welcome Dr. Mohammad Afzal Azam sir to give a brief introduction about the webinar. Sir, just a webinar, sir. Then I will call the.
Very good morning to all. On behalf of JSS Academy of Higher Education and Research, JSS College of Pharmacy, our faculties, we welcome you all participants for this webinar. My special welcome. I also welcome to Divya, our eminent speaker, distinguished speaker of today's webinar from Altem Technologies. She is going to deliver about the basics and little bit knowledge about your all technologies, all molecular modeling tools, whatever is there in their package. Now, participant, this is very important nowadays to learn this technology, to learn this your softwares, so that you can excel in your research work. So nowadays it is required everywhere, whether the industry or research or academia, everywhere it is required. Without this, you will not be able to publish a good paper. And now you imagine the current situation, how it became very important. So you can discover drug in very fast, in very short time, and with less expenses using these molecular modeling tools. So this BIOVIA, that drug discovery suite, it is very important, okay, very and you can say reliable software where you can do your work. So with this, once again I welcome Dr. Divya, Senior Application Scientist from the Altem Technologies. And I request Dr. Kali Rajan to just introduce Dr. Divya and proceed for your webinar. Dr. Kali Rajan, over to you.
Yes, sir. Thank you, sir. So good morning, respective delegates and my dear colleagues. Introduce Dr. Divya S. Link 3D printer. She was BIOVIA discovery student.
I think, sir, the connectivity, sir, we lost you.
Dr. Kali Rajan, you are not audible. Just Kali Rajan.
Yes, sir. Yes. Now it is audible, sir.
Yeah, it's
Yes,
yeah.
So Dr. S. Divya is expertise in molecular modeling of proteins and transmembrane, structure and ligand pharmacophore based drug designing, and molecular dynamics and simulation of lipid systems. She has more than 30 publications with his credit and 3 book chapters published in New York Science Publishers, USA and FUA. She is also member of Indian Science Congress. Previously, she was worked in as an application scientist for life science, Apsara Innovations, Bangalore, and also worked in DSIR lab and Research Foundation for USA collaborative project in Chemoinformatics. She is also advisor for BioName Department of Bioinformatics, Women's Biotech Park, Chennai, and she was a trainer for various bioinformatics courses and Accelerate Discovery Studio. She was completed a PhD in biotechnology in computational pharmacology and cell-based assay against various drug targets of cancer. She completed MTech in biotechnology at Satyabama University. She got a gold medalist in MTech and a BTech completed in Anna University. And she has more than 50 plus invited talk in bioinformatics in various reputed institutions. So with this brief introduction, on behalf of our department, we welcome Ms. Dr. Divya S. for this live webinar on state-of-art molecular modeling tools, BIOVIA drug discovery suite. So thank you, madam. So you can proceed.
Dr. Devia, that our participants are eagerly waiting to hear from you.
Definitely, sir. Definitely.
And it will be very useful talk for all our participants. So
Definitely, sir,
I
definitely.
request you to start.
Yeah, thank you so much, Dr. Kalirajan sir, Dr. Afzal sir, for brief introduction. With this, I can't wait to start my session. I'm starting my session, and participants, I welcome you all once again from JSS College of Pharmacy and Altum Technologies and BioVia Discovery Studio. If you have any queries and you want to discuss it, please kindly let me know. I'm very happy to and delighted to answer all your queries. Put it either in a chat or you can raise your hands and we can discuss it. So whatever, I think this is going to be a very comprehensive session and lots of concepts I'm going to today deliver. So whatever you have, queries you have, feel free to reach to me. I am delighted to that. Okay? Delighted to answer all your queries. With this, I start my session. I'm sharing my screen. One second. Yes. So I'm sharing. So BioVia Discovery Studio software. Earlierly, this software is called as an Accelerate Discovery Studio. So this is a more than 3 decade software, has a various imprints in technology of drug discovery process. So whatever drug discovery process that is you want to perform, say for instance, I want to do certain pharmacophore, I want a ligand base, I want to perform small calculations, library designing, reactions, I want to perform whether my reactions will go for the synthesis or not. So for all kinds of solutions that people will address in pharmaceutical domains and life sciences domain, biological domains, for all people, this is a one-to-one end solution software. So today I'm going to take upon the topics which is going to cover overall about, for example, you want to publish in very high impact factor journal and your research need to be recognized and it should be in a high index journals. We should prepare in such a way our screening process should be more reliable, and when we take into in vitro or in vivo testing, we should have more reliability to give you the perfect results. So that is what I am going to discuss it today, bridging your in silico and then in vitro and how you can perform using art of modeling and simulation Discovery Studio using a software called BioVia. Let us start the session and keep walking along with me. About our company, Altum Technologies is a more than a decade company, have a various imprints in 3D printing and scanning technology. Now they are also into a 3D bioprinting technology. One of the technology, one of the domains or a vertical is about BIOVIA licenses, the BIOVIA Discovery Studio, where we are dealing with a drug discovery process, and we are also into the material research processes. And you can see esteemed awards of Altum Technologies on your left-hand side corner. So, coming directly to BIOVIA. BIOVIA is a rich scientific portfolio software. It starts from a unified lab management and ends with chemical reactions and formulations. And if you want to see, for instance, in a unified lab management, people will try to replace. Now everything is becoming smart works, and people are trying to put everything in your smart calculators and smartphones like that. Same way, you are also having an electronic laboratory notebook and connecting with the cloud computing, where you can store your data, where you can download your data wherever you want to perform. And maybe a year before or a decade before, 2 decades before you did the experiment, when you're saving in this particular lab management, you can download and you can analyze it at any time. And of course, if you have any software or a very company which is dealing with productions and management, we have a process production operations, and then we'll be dealing with dynamic mapping, authentication, conceptualizations, and then dealing with all kinds of data. And when you have your data, when I say about a data, we definitely will go for a quality and regulatory management. Whatever the data that we are getting, that has to be managed. Even in a pharmacological domain or drug designing and drug releasing also, there will be a drugs will go for a quality control and quality analysis. Only then it comes to the market. Same thing for the software also. We'll be more giving the importance to quality and regulatory management, where we'll try to document, process, and compile and finally go for a collaboration. When I say about the collaboration science, the collaboration science, we have a solution for all kinds of problems. Say, for example, you're having a chemical compound, maybe say you have curcumin in your hand. We know that curcumin is known for a various of pharmacological activities, but you want to discover something new from that. What you have to do? Just take your compound and connect with the different biological drug targets. So just input of your compound, it will say you what are the drug targets are there. So because basically the software has been integrated with the artificial intelligence and machine learning algorithm, will help you to predict and analyze the data perfectly. And novel therapeutics, you are coming to know about an antibody-based drug design. There are a lot of antibodies are made and have been utilized for various diseases control. Example, cancer, HIV. So like this, we have a novel therapeutics. So we are also having a domain to discover your biologics, large molecules, antibody designing, optimization, and studying as a function of a temperature and aggregation scores. That's also a part of a discovery software. Then once you design your drugs, or you want to delivering a drug is a major task in a pharmaceutical domain. How we do that? We need a certain chemicals or materials like a polymers or a liposomes or a made in a nano-sized particle using silver or gold. These material feasibility need to be analyzed perfectly. What is its property, whether it will deliver or not, whether it will make like a measles or not. So that kind of an analysis, choosing a drug to delivery approaches based on can be done by using material schedule. And coming to chemicals and formulations, you have maybe you want to develop one formulation for certain products, and that product, for example, you want to develop a cosmetic product. In the cosmetic product, that product should be free from carcinogenicity, mutagenicity. It should not make your skin irritation, ocular irritation. So like this, there are various models that need to be analyzed. We can't do this by using our animal trials or maybe human trials. This is definitely will consume a lot of time and money. Instead, what we are going to do? We are going to take those chemical molecules and we are going to check it out using artificial intelligence-based algorithms. Like, for example, if I take about tell about the machine learning algorithms, Bayesian categorization or supervised learning algorithms and unsupervised learning algorithms. So like this, I can use a combination of an algorithms and I can study the properties, whether it will make skin irritancy or not. So for formulating a certain product, we must need an including your computational works will help you and ease your process. So for all these kinds of solution, we are having the solutions in BIOVIA Discovery Studio software. Directly getting into a topic, this is about the scientific portfolio. Collaborative science, I explained detailing. So we have more into life sciences, material, research applications, chemical inventory management. And if we take about the life sciences, biologic modeling and simulation, small molecular modeling and simulation, and QSPR workbenches based on machine learning algorithms. So all these things are part of this BIOVIA Discovery Studio software. So what we are going to do today, why we have to study this process and why it is required. You might have been thinking about it. Say for example, if you're a chemist or a pharmacologist or a pharmaceutical chemist or pharmacologist, whatever the domain, ultimately we are going to start with the chemical compounds. Okay, you have chemical compounds. How do I say among them you have a lax? Maybe I could say that you have a lax of molecules in your hand. With that lax of molecules, which one I will pick? Which one I have to say this is a potent molecule? Can I do it? Okay, maybe if I take a traditional technique, I have to take a molecule screen. So that choose screening of 1 lakh of molecules is not an quite easiest process. It will consume lot of time and you have to energy, human intervention. So it will make your work hasten. Rather, when I think about it, why don't you see everything has become automated and then the information technology is giving you more hands to us? Why don't we introduce chemistry with informatics? That's what chemoinformatics is arrived. The chemoinformatics is basically information technology is playing a major role in the chemistry domain and use of this technology to solve various problems in a chemistry. It also includes quantitative structure-active relationship. If I remove Q, SAR relationship is known for more than 6 to 7 decades. And when I say about when I remove S here, quantitative structure property relationship, each structure have its own property and have a relationship with the activity value also. So which I can do it by hand or can I do it by means of small mathematical calculation? Definitely you can do, but it consumes lots of time. I want today discovery process, tomorrow I want a results. Day after tomorrow I want to test it in a wet lab technique. How it is feasible? Is it feasible? Definitely it is feasible. It is not at all a magic. It can be done. You have a lakhs of molecules, you can screen overnight and you can draw a conclusion within a day and next day after tomorrow, within a week, you can test it in your in vitro testing by utilizing your consecutive protocols and follow the proper workflows in computational biology and chemistry. So this chemoinformatics also deals with the pharmacophore properties. What is this pharmacophore? No issues, don't worry about it. Each drug have its unique pharmacophore. If you remove that functional group of your molecule, that property of the molecule will be lost or it might be reduced. Sometimes it might be increased. So that means the specific functional group of a molecule attached to the parent molecule is considered as a crucial or essential pharmacophore. That also we can predict. So this, if you have all the solutions, then it will be a fruitful boon to your pharmaceutical designs, right? So pharmaceutical company will definitely use this. Of course, lots of companies are utilizing this technology. How come the vaccine development has happened within a year? Think about it. We are using various advanced technology in artificial intelligence and sticking out with the vaccine development. That's what we are arrived as soon as within 18 months, we are arrived with the vaccines. So definitely for screening of drugs also, maybe next year or a coming year, we will definitely have a solution for SARS-CoV-2 also. There is no doubt in that. Even after if any pandemic arises also, we no need to worry about it. We are having such a tools and advanced technology to handle these situations. So this is about an objective why we need to go and study about the chemoinformatics and drug delivery, drug discovery process. Okay, so that today we are going to see this only workflow, how to bridge. Imagine you are having a molecules and you are going to screen high throughput screening and then virtual screening technique, molecular dynamics and simulation. No worry about it. Molecular dynamics simulation will fall on your hand when you are going to use this discovery schedule. Free energy calculation, no issues. It is very easy. You can calculate delta G by clicking one point and you have arrived at the solution. Finally, okay, starting from millions of molecules, finally with a few numbers. With that few numbers, you can with confidence, you can go for in vitro or in vivo testing. If not, on the other hand, what you can do, already published molecules will be there. Test with this flow and then see and connect with your study and then make your benchmark, then start your research process. We have tools to do that process. So we are going to see each and every step in detail and why we need to do this process and what is its application. That's what we are going to discuss it today. So next 1.5 hours, we are going to be on this particular box and going to gain this knowledge and whatever things are falling in your mind, please put it in a chat. We will discuss in a Q&A session. Thank you. Now starting this process. Now imagine I have a library of molecules. I am getting now into one virtual screening process. Now I am getting into this process. So ADMET, when I say ADMET, okay, you take a drug that should have good absorption, distribution, so we can see now. How the top cat, toxicity profiling using animal models? Yes, we can do that. Pharmacophore screening, similarity based. So first I am going to deal with this. Then I'm going to step inside. I'm going to funnel it very densely and going to identify the compounds. That is my overall workflow and that is the objective I am going to do it today. Let's walk along with me. Virtual screening. If you are new or already exposed to this technology, I'm telling once again, virtual screening is basically a computational method. So I'm going to deal with a computational method to analyze a large database of compounds. So not 1, 2, 3 or hundreds of compounds, lakhs and millions of compounds we can analyze and to identify the potential hit candidates. Very important when we are spelling this word hit, lead, drug. All of both three different things have a different properties. Okay, I will discuss as keep moving on. So that can be done. Say you are having maybe you don't have a library. You are downloading from Asterix or Zinc, or you from PubChem, or you can say that ChemDiv. Lots and lots of corporate libraries are available. So you can download from them in your various chemical formats, SD, STF, or whatever the chemical format is widely acceptable in Discovery Studio. Visualize it as in the software. So this mode of technique, if I can do my research for 1 lakh of compounds per day, that will definitely have a crucial significance in your lead discovery process. Moreover, it is very quick, economic method to identify your novel active molecules. And moreover, you know it is always sit on a computer. Automated process, select, click and go. It will be automated. More cost effective and saves your time during research. So this is what we require in this fast-paced world. We want to make everything automated and make everything very cost effective. I want to spend only around, I could say that 10 or roughly I can say $1 million to discover your drug. Yearly, they will take $770 million for one particular drug to come into the market. Now it has come to 1 million. Of course, if we are going to reduce so much of costs for discovering and then taking this kinds of drugs by utilizing the smart software like a BIOVIA discovery. So this is why we are on the virtual screen. How we are going to use this? Different types. What type I can use? You can use any approaches. That's what in a research there are two things always falls in your mind: yes or no. Flow chart will goes like this. Yes, okay, I can go ahead. What happens if no? So that's why we are having a multiple approaches. Either you can work it on ligand based or you can work it on receptor based. Some people who is strong in biology, they will go for a receptor based. Or some people who is strong in chemistry and pharmacology, they will go for ligand based. What they will take a structure of compounds and they will retrieve and see for the shapes, complementarity, substructure. Because what we will see, same similar like structure will have similar like behavior in the system. Same concepts here also. Shape, all these things they will study and they will go for an analysis. On the other hand, receptor-based screening, where the people will try to take a protein or a drug target protein, and with that drug target protein, they will try to understand the docking sites. Maybe you are giving a blind docking. Blind docking also can be done by using Discovery Studio, using a C-Docker algorithm. You can do global docking or you can do specific docking. Whatever docking you want, yes, of course, we can do that. Okay. And then this process can be repeated several times and for a post-processing approaches, and flexibility of your receptor and ligand also can be done. Even you can do flexible docking for both receptor and ligand so that both will be in motion, and with that also you can analyze your results. So both ligand and structure. Sometimes you can combine the both if you require. This is the types of digital screen technique. Okay. If you see certain things, definitely there will be a pitfall. Any technology that they're using that will have an errors. So then what are the errors that we have? It always falls in the four major categories. There are still lots of things, but when we try to integrate, it will fall under these categories. Erroneous assumption expectations. Whatever target what you're taking, that might be from a different organism or something like that. And data designing and content, choice of software is very important. Reliability of algorithm is again very important. And then sampling of your ligand is again most important thing in the ligand based drug designing, and to understand the target flexibility. So all these things all need to be analyzed, and then we have to draw the conclusion. Without that, we cannot draw any conclusion in that. So let's see what each and every one in that. How to overcome from this pitfalls and to execute a strong drug discovery process and to have a series of fruitful results. Implementation. So something if I say in words also, it will not reach easily. So that's why we always should have some implementation using workflows like this in a kind of diagrammatic representation, chart-like representation. Implementation. See, for example, I have divided into three different things. One is by means of ligand based, one is by means of computational based, one is in the structure based. In a ligand based, I can choose a pharmacophore model. What is this pharmacophore model? No issues. I will tell you when you keep walking along with it. In the pharmacophore model, we have, say, for example, curcumin. Just take an example of curcumin. So I know the curcumin is very good. So for that curcumin, I am designing some, I'm trying to create a feature of pharmacophore model. And with that particular model I develop, I can screen a set of ligands or compounds. So that using a particular protocol called ligand pharmacophore mapping. Then I'm selecting. Well, how we select
130 participants.
or strong with
Divya and Takayoro.
selected based on the fit value. Then screen. And then once you identify more strong and fit based, so for example, your curcumin is having 10 features. All the 10 features are fitted by one molecule, that is called the best fit, highest fit. Or for example, you are taking a molecule that can match only with the 5 features, 50% fit value. So like this, you have to select your compounds based on the fit value. Then it will get finished, because once you screen, you will have a lots and lots of molecules will be there. Still, it is not cost-effective process. So again, we are getting into virtual screening technology here. When you go into this process, we are screening for ADMET process, checking for the ADMET, and then animal model testings, and then drug likeness. Then we are going for a high throughput screening technology using molecular docking and optimization, and molecular dynamics and simulation, free energy binding, energy analysis. Okay, I don't know anything about my ligand. I don't know anything. But for example, when the pandemic has arisen, people were landed up only with the protein molecules. That means searching, doing a homology modeling technique, and then they identify the model, the similar like structure for SARS-CoV-2. With that, can I go for a drug discovery process? Yes, of course. That technology is called as a structure-based pharmacophore-based modeling. So you know the protein. If you don't know the structure, active site also, no issue. Any protein will definitely will have cavities. That you know, the bigger the cavity, the more probability your compound will go and bind. So considering that concept, we are taking the structure and then we are designing the pharmacophore. And after designing the pharmacophore, we are again going for an ADMET screening, TopCat screening like that. So there is another scenario. I'm not going to use any of this technology. Rather, I have only the compounds. I am a medicinal chemist or organic chemist. I have only lakhs of molecules in my lab. I want to screen that process. How do I do that? How do I do that process? Of course, you also have solutions here. Take your molecules, go for ADMET, TopCat, drug likeness, docking, and then finally arrive the solution. With that solution, you go for an in vitro or in silico in vivo testing. So we have a solutions for all the problems. Now we are going to discuss how to implement each and every step in detail. So I told you, right, no need to worry about pharmacophore because I'm going to discuss it. Pharmacophore is nothing but a structural ensemble of the compound which is required for your biological activity. Either removal or replacement of the group will increase or decrease the biological activity. This is a simple concept for our pharmacophore. Anybody can define it. Okay? So how you can design your pharmacophore? It can be qualitative analysis or can be done by quantitative. What is a qualitative? So qualitative, you are not using any biological activity value. Rather, you are considering only the compounds. That is called common feature hypothesis. Even I cannot design as such, right? It needs to be done by using a sophisticated or a very reliable algorithm. Catalyst hip-hop algorithm is known for more than again 2 to 3 decades, which has been part of a discovery studio, which can be helpful to analyze and to identify the common feature in your compound. And even if you have set of active ligands, you can be give there and you can validate or for an ROC characterization, recursive operating curve characterization. So you get a AUC curve, area under the curve. From the area under the curve, one can predict the results. Or in contrast, we have a quantitative analysis, that is structure based. For example, you know the quantity. Maybe I'm working for a certain cell line, maybe HEPA-G2 cell lines. I'm working on that. Previously, some people worked on, and I want to take that results. Can I do the discovery process? Of course. So the method of retrieving the previously done results for specific cell lines or drug target or assay or enzyme, and then utilizing and developing a model based on that is called PDQSPR. It will help you to identify the features or a crucial functional group which is required for the biological activity value. So without going blind, it will tell you about the features which is required. It will plot the features based on the pharmacophore. That will fall within the PDQSPR pharmacophore generation. So now I am going to concentrate more into that how to generate your PDQSPR pharmacophore and then how to pick the pharmacophore for screening of the molecules. Now gets into the 3D QSPR. 3D QSPR can be called in different names from quantitative pharmacophore model like that. All the pharmacophore that we are developing are energy based. Along with energy, we are using catalyst conformations, and then it's a hypogen algorithm. Let's see how this algorithm is going to work. So 3D QSPR is a protocol to generate your predictive pharmacophores and developed by means of catalyst hypogen algorithm and published by Lee et al. In 2000 to derive a structure activity hypothesis model for sets of known ligand whose activity values are very important. When you see papers or come across this word, then definitely you can blindly put that is based on activity value. So how it is typically different from QSPR, because we can read a QSPR by means of machine learning algorithm. So this is different from typical QSPR. In the QSPR, we are mostly concentrating on descriptors. A log T, acceptor, donor, electronic, topological, surface area, molecule. So like this, we are dealing with the various descriptors. But rather in 3D QSPR, we are dealing with an alignment and fit of the pharmacophore model rather than the molecular features. So this is the difference between the 3D QSPR and the QSPR model. How to generate, how to use this technology. So behind that, we are having a hypogen algorithm. How this hypogen algorithm? Say, let us consider or put a case like that. I am developing a model for ATG2 liver cells. Okay, and then other person, maybe an other set of people in the lab, or no need to test once again. Maybe previously my senior or a super senior or a person who worked in laboratory has data. With that data, I am going to generate a model. With that model, I am not all the compounds, maybe hundreds of molecules are there, they have tested previously within over years. And then again, I am not going to test my molecule. I am having thousands of molecules here. I cannot test all the thousands of molecules in HPG2 cell lines. Rather, I can prioritize, or I can top the 10 molecules based on the mapping. So how this hypogen will construct a model is very important. So as soon as you give a ligand along with your biological activity value, this will process under the hypogen algorithm. So first it will construct, then it will subtract, and then it will optimize. Once you go for constructing, immediately your ligands are placed, and then common active molecule will be considered first, and then that will be in your set. No need to worry, don't think about test and fail. Any set of the molecules, it will try to generate your actives. Presenting your actives, it will try to construct the pharmacophores. Then subtracting. Whether that pharmacophore as constructed is correct or not, will be useful or not, that a subtractive phase will take care. And then if any feature is mapping with an inactive compound, maybe one component 0.1 nanomolar, my last molecule is 7,000 nanomolar, that is considered inactive. If the pharmacophore of active and inactive are matching, it will subtract. It will delete that particular pharmacophore. Then it will go for optimization using simulated annealing. Then finally you get a model. Now let's see this each and every phase. Constructives. So we try to identify the actives and put the actives like this. And mark the pharmacophores, the acceptor. So something you see it in green in color is acceptor. It's a definition for a color definition for a pharmacophore. And then donor. And then you have a hydrophobes will be cyan in color. Ring aromatic will be orange in color. And negative ionizable will be in a blue in color. Like this, it will have various abbreviations, color-based abbreviations. Now I place this active one. Now the algorithm will put the active one here, and then a second active one here, and see the features. Okay, more or less similar, and try to extract those features. This is the importance of constructive phase. It will make it like a beans. It doesn't know anything. It knows only to translate. We studied by translating the languages. So now we have a constructive phase. Now having a constructive phase, now we are getting into a subtractive phase. Now the last I told you, there is one inactive molecule. It will try to take that inactive molecule. Place that inactive molecule in the constructed one. So it will try to place the inactive molecule in the constructed one. In case any pharmacophore is matching with the active one, it will subtract. Can you see this? This is a constructed using active. Inactive is matching here. So it will subtract that phase. So any feature which is mapping with the inactive must be subtracted. That's what this algorithm will do. Now the subtractive phase is finished. Now it has to be optimized. So optimization, it will try to create a hypothesis and estimate the quality and complexity, which is having a more R value will be taken on this. So like this, it will try to put together and forms your using a simulation. It will move here and there, catalyst, so your ligands will be in a motion, interface distance, everything, each and every parameter has its role in a pharmacophore generation. And check it out. And finally, whatever energy you are giving, 5 kilocalories per mole, 10 kilocalories per mole, 20. So if Discovery Studio software has a feasibility of a user to do or discover various processes, you can estimate or can pick your ligand what you want rather than the default options. So finally, we'll be getting a pharmacophore generation by rotating a vector, translating into a random selection, adding a remaining feature, changing a relative weight and tolerance to improve the scores. And optimization, it will try to use an RMS, correlation coefficient, and low total cost to be identified and take the top 10 scores. And it also uses a Hoffman-Draser principle to do this process because it's the simplest hypothesis accurately estimates your best molecule. So like this optimization will keep on moving. And finally, we'll be arriving the results. So calculating a fast geometric fits using a fast ligand pharmacophore based algorithm and also performs a linear regression model versus geometric fit to estimate your activity of tested train set ligand and generate a geometric fit and predict the weighted activity value, and finally a statistical parameters including error cost, weight cost, configuration cost. So like this, I'm telling all the stories, right? All the things the software will take care. Finally, we get only this out. That means Fisher validation. So one of a validation method, internal validation method of a pharmacophore model. So it will try to validate a model, and then Fisher validation will say how much significance of hypothesis 1, 2, 3 like this. And automatically, if they give the validation internally, you get a Q square value also, which will be plotted like this: log of activity versus your observed activity value. So more linear and higher the R square, better the results are. So like this, first confidence level, okay, fine, good, I'm taking that. Q square value equivalent to your test and training, yes, of course. Test and training can be validated internally over here also. You can do externally also. Either way, you can perform this process. So I'm taking a pharmacophore 1, 2, 3, like the 10 models are there. I'm going to test it. This is how the results will be generated. So you can see the features that has been mapped: acceptor, donor, hydrophobic aromatic, hydrophobic, and positive ionizable. Tolerances, total cost, correlation, 0.98. Very good correlation. And less RMS. Yes, good. RMS is also good. Root mean square deviation is also good. So as you keep seeing that, root mean square value will be keep on increasing and the correlation value will be keep on decreasing. So usually hypothesis 1 will be good, but with that I cannot draw the conclusion because still more mapping parameters also need to be considered here. So Fisher validation, and I'll get a pharmacophore summary. And then I'll be getting a random sheets with a cost value. That will be created random sheets because 90% confidence level will try to create a 9 random sheets, and along with the correlation random sheet. All this ones will help me to see if there is any zeros or there is a deviation in your, always your correlation will be top when compared with other random sheets. Always your cost will be lower compared to other random sheets. This is how we have to interpret the results. And finally, we get a hypothesis along with the error and estimates. And the hypothesis will be created. You will get a fit values, total cost correlation. So majorly, what parameters we have to analyze? Correlation, RMS, total cost, null cost. Null cost is always away from your results, and total cost is always towards your results. Okay, upper limit and lower limit, like that you can see it. Okay. If the plots will be given, you can see the status, estimated activity value, error value, mapping. So you can see the mapping. There is one star. I keep moving on, and then you have to take in a top 10 scoring analysis. So we'll be getting a total cost difference. So in case, since it is a wide and comprehensive session, I cannot cover in much about what is the total cost because we are having a definitions for that. So see our BIOVIA Discovery Studio 2020 DDH YouTube video. There we are explaining in detail about what is the total cost, what is the cost difference, what is RMST, how the correlation. All the features we have been explaining in very detail. If you have still more queries, you can get back to us to the inquiry@alton.com. So null cost. So I am trying to see that null cost will be always apart. Null means zero, right? Same thing, zero cost. Null cost, you can think like fixed cost is always fixed. So from that cost, how is the total cost is calculated? So that's how the hypothesis will be calculated. Fixed cost, configuration cost for your 3-dimensional spaces, and then the cost difference is always from a null cost to fixed cost. Always are higher, comparatively higher, and your values are towards the fixed cost is considered best. This is a statistical analysis. Now, I pick actually 2 different hypotheses. This is again a task. I have 2 hypotheses which is generated. Which hypothesis I should pick. So I told already the correlation because having the best is considered good. Of course, I will totally agree with you. Now I am seeing that there is one star in this mapping. Okay, what does this star means? I am taking another hypothesis. There is no star. The star indicates there is an inactive mapping. So what I should do, I should take hypothesis which is not mapped in the star in the active part. If I find a star in active, I should omit that hypothesis because there is maybe a false positive might occur when we are going with in vitro testing. So I have to be very careful when I see the star. So when I see the star, I will omit this hypothesis. Rather, I will take this hypothesis. Okay, now for HPG2 cell lines, I have generated the model, or MC7 cell line, I generated the model. Now my laboratory, maybe JS's laboratory or some college laboratory has developed this model, can be used internally to screen your any library of molecules, and you can name this Pharmacophore model along with your college name or who developed this model. So with this model, for particular model, I can screen my ligon. I have an output Pharmacophore model and I mapped it. This Pharmacophore model, this CERMI has a standard for screening your diverse set of Pharmacophore, diverse set of ligands for HPG2 cell lines or MC7 cell lines based compounds before testing into in vitro. This is how far it will take, say, taking 1,000 molecules, 1 lakh molecule and testing. Rather than I can screen here with this particular model, I can draw my conclusion. So which one is very easy? Please think about it. Taking 1,000 molecules for in vitro testing or screening with a model, developed a model using 3D QSPR and prioritizing 10 models for in vitro testing. It's feasible. Think about it, which is more easy for you all, but little bit work you have to do, not more than 2 or 3 days. If you are new person, it will take not more than 2 to 3 days. If you're already expertise in that, it will not even take 2 to 4 hours to complete this process. That is the difference of working with a cost-effective manner and more reliably using the software like this. Now the first concept is completed. Now I have mapped my molecules. It's ready for the process. I have taken millions of molecules. I end up with a lakhs of molecules. With the lack of molecules, I am getting into the next process, ADMET process. Okay. In the ADMET process, still I have lakhs of molecules. I cannot go for in vitro testing. That's why I am screening my molecule. Till Pharmacophore, I call my molecule as a hit, because only when your molecule is going to obey pharmacokinetic and dynamic properties, it is called as a lead-like molecule or a drug candidate molecule. To understand or to name like that, it should obey the ADMET and TOPCAT properties. That means your drug should have good solubility, should penetrate a blood-brain barrier, and should not inhibit your metabolism enzyme that is present in the liver, and should not cause your toxicity to your liver cells, and should have a good absorption, and then should not bind to your plasma protein binding like albumin. So all these properties I have to analyze. Can I do it by in vitro? No, it is not feasible. It is not advisable for testing the lakhs of molecules. That's why I'm going into a computational based pharmacological analysis. So how do I do that? This is Discovery Studio client page, licensed version. You have a tools for macromolecules, pharmacophore, receptor ligands, simulations, library design, analysis, end-to-end solutions for all kinds of drug discovery process. Now, I have a lack of molecules over here. With that lack of molecule, I'm selecting the 6 different descriptors or models and going to sort out my results. You can see here 489 molecules done in 8 seconds. Please kindly calculate. 418, approximately 500 molecules in 10 seconds. So if you have lakhs of molecules, how long it will run? Not more than an hour. And then when I click a plot S, of course I get a plot S. And then sorting the results is also very easy and binary method. See, solubility, there is a software, they will also tell you the levels. This is a level is good. 3, 4, 5. Consider as a good, bad, and then moderate. Similarly, blood-brain barrier level 0, very high penetration, 1 is the penetration high, like a false. True means is binding false, like this we have a categorical data. And then continuous data. So 6 parameters, 6 levels, everyone can easily understand. That's why we are sorting our results based on the levels rather than numbers. So we have that both the data will be created because user will have a different perspective. That's why we will given a more data and results. Which one you want to pick, that you have to decide. I am choosing always, it is very easy for me to understand. That's why I always pick based on the levels. Level first solubility. They are giving based on the relationship that are predicted, quantitative relationship using in a Journal of Medicine they published. Using particular data, they develop a model and then they are told that at solubility level 3 is good, 4 is optimal. And we get a plot will be plotted like this against the state of lipophilicity, A log P98 versus the polar surface area of the molecule. And then I'll be getting confident levels. This is called optimal prediction space. The molecule which is falling under this optimal prediction space and statistical level will definitely obey ADMET properties. That is in a blind idea. So absorption, 99% confidence level, absorption 99%, blood-brain barrier 95, 99 like this. Okay? Predict aqueous solubility at 25 degrees Celsius. Now I am taking only the level 3 and 4. If I select only the level 3 and 4 lakhs of molecules, I will end up only with this. Almost 70% it has gone. 60 to 70% I lost the molecule. If I take among one of the molecules, when I go for in vitro testing, what will happen to me? Definitely I will end up with the problems. Again, I have to restart my research. Since I don't have funds, all those things, it is not appreciable, right? So that's why we are doing this analysis using in silico or computational software like Discovery Studio. Now, blood-brain barrier screening, whether it is causing blood-brain barrier or not, check it out. Take the level 0 and 1. Wherever other than the 0 and 1, delete it blindly. Okay, because it's known and developed based on level 0 is very high, 1 is a high. It predicts based on fast polar surface area and countable and base logothermic value and calculate the ratio. Fine. Now I take only these two. See this? I lost some of the molecules. And CTY, it should not be inhibitor. False is non-inhibitor of this particular enzyme, and true means it's inhibitor. So I'm taking only the false. Next, hepatotoxicity should cause toxic to my cells. So I'm taking a hepatotoxicity non-toxic. False is a non-toxic. Okay. I'm taking only the false. Finally, coming to the absorption, I'm taking only the good absorption level and the moderate absorption level. Okay, I'm taking few more. Last, plasma protein binding screening. So predicts the how much binding to the albumin or plasma protein binding. I end up only with this. I started with millions. When I come to 3D QSPR, it's a lakhs. When I come to ADMET screening, I have only few hundreds. So imagine how much efficient. I have only few hundreds of molecules. With this, my process is completed? No, still it is there. We are going into before ADMET screening. After ADMET screening, you can see I have only 46 when I take 25,000. So calculate accordingly. Now, getting into now, I want to test my models in animals. I have two different models. One is these two are a machine learning based algorithm developed model. That is, one is by means of classification method and another means of regression based methods. So classification method says whether your molecule is carcinogen or non-carcinogen. And then my regression based method will say what this could be a set dosage for this particular compound. Okay, along with that, they use a QSPR technique. So far, I told initially QSAR, QSPR, quantitative structure, property, and activity. Now we are getting even into toxic relationship along with the machine learning algorithms. So classification model, it will tell you using a Bayesian analytic techniques, carcinogen or non-carcinogen. And regression models will tell me about whether this particular drug or can be a therapeutic compound, or you can whether it can be used for the accessing for a commercial wise or industrial wise or not. What could be a dosage for animals that will help me to predict. So doing this model, I take the model compounds after ADMET screening and testing for an FDA-based food drug approval or national toxicity program based. I will pick the model, or I can whatever model I want. You have two environmental models are there: aerobic biodegradability, skin irritations, ocular irritancy. Like this, you have an extensible models. If you want to test in multi-site means you can go for top-cut prediction. Extensible is the basic one. Okay. In depth, single carcinogen site, multi-carcinogen site, you can go for a top-cut toxicity prediction. So we have a various models in that. Select the models. See the similarity. Why we are giving similarity? Even for activity also there is a similarity. For the toxicity also there is a certain fingerprints are there. That's why we are giving a similarity search. And then detailed report to create a report in a detailed format. Select the model, execute the process. Finally, we'll be getting in what could be a molecule, whether it is a mutagen or non-mutagen. Mutagen means how you believe this is a mutagen. There is a fingerprint says this is a molecule to be a non-mutagen. There is a fingerprint says this molecule is a mutagen. Position of the molecule or atom have a greater role in your carcinogenicity as well as for activity. In case the specific position, say for example, this position is going for a toxicity, you can remove this functional group and replace with some other functional group, test it and check it out whether it is causing still carcinogen and mutagen. Make your molecule as non-mutagen, free from a carcinogenicity and mutagenicity. That is very, very important for you any kind of lead or a drug molecules. You can test it in various animal models like a mouse female, mouse male, rat mouse, rat male, rat female. Like this, you can test it with various animal models in Discovery Studio. See here, you will have a mouse model. You can predict TD50 also, thermodynamic dosage rate, and fat at minnow, rat AC50 value, rat TD50, rat oral LD50, rat inhalation LC50. All the things you can able to predict it, and you'll be end up with the solutions. So finally, now imagine lakhs of molecules. From the 100, I have only 50 molecules in my hand. Again, I'm testing now. This is for drug likeness. Whether my drug will have oral bioavailability or not, that I am going to test by using Lipinski's rule of 5 and Webber rule of 5. So these two peoples are more important and define the rule for your drugs to be have a good oral bioavailability. So strictly follow these rules and then you can draw the conclusion. Now after that, I'm having only a 30 molecules or 20 molecules in my hand. So that I will prepare using one prepare ligand protocol to add hydrogen, calculate coordinates, ionize function group, remove duplicates. All this process I'll be doing using a small molecule tools and prepare ligands. With this, I will go for a prepared set of molecules. So finally, I have only 20 molecules in my hand. Started from millions, lakhs, screening within ADMET properties, shortcut properties. I have only 50 or I could say the 20 molecules in hand. With the 20 molecules, again, I can go for an in vitro testing. Still, it is not economical. I could say that people who don't have funds, it is not much economical because each and every assay is cost more than 1,000 rupees. So 22 molecules, 22,000 for single assay, still most considers a huge thing. So, but that's why we are going for high throughput screening technique. So we are screening still more, funneling down our results. It's not completed. We are still going ahead. We are densing, funneling down. That's what we are using. High throughput virtual screening technique and docking technology. Now get it. So what is the concept we call as? This is called structure-based drug designing. So you have to you should know what should be your protein of interest or drug target of interest. In case if a drug target is not found in your structural database, no issues. Still, you can model your structure using homology modeling using modeler algorithm and discovery. Okay. Prepare your protein. Okay, preparation is done. Preparing your protein because during extracrystallography, a lot of problems will arise that has to be addressed. Define your site. Prepare the set of ligands, which we already finished. Choosing a docking algorithm, very, very appropriate. Analyze your results. All these things we are going to do now. So validation, very important one. I will say that docking is not a simple process. Taking a compound and doing docking. If you are taking your bound ligand, do validation. Set up your parameters. Always your docking, doing a redocking should be less than 3 angstrom, extreme level. Okay, less than 3 angstrom should be RMSD of your docked ligand. Then only, otherwise you can optimize the parameters of your docking. There are a lot of parameters will be there doing while you're performing docking. Don't always take a default parameters. Try to do research in the parameters and define your own parameters for your drug target. That is how the productivity of the research will be there. That's how we should do research. And check that, and whether you're changing a single parameter, 2 different parameters, it is reducing your redocking process and you're getting RMS less than 2.5. It's very good, ideal thing. So that you can take up your compound or known compound which is screened and do docking and then analysis. That's what validation is very, very important. Okay. And there is again screening your molecules and prioritizing. So we are going to prepare doc scoring function. This is totally dependent on the which algorithm that you are taking. There is no standard scoring function. There is no standard for your molecule. Your scoring function should be like this, should be this values. There is no standard rules at all. Depends upon your molecule and then a protein, and as well as the scoring function, it is a variable parameter. Now, protein preparation. Once you prepare your protein, download your protein, it will show all the problems. What are the invalid residues, or the incomplete residues, loops are missing or not, all the details it will give. Once you click, you get this report. And also active site that will also load up the keyword, whether you are either there is any hydrogen bonds or any water molecules are there in active site or not. All the details you can able to get it when you click the active site. Now there is also alternative conformers will occur during your process that has to be removed. Once you click prepare protein, this will take care of all the process. remove water molecules, unnecessary water molecules, standardizing atoms, getting alphabetic conformers, modeling your loops, very important because loops or the regions will keep on moving when they do. It will not stand in single position when they do X-ray crystallography data analysis. It will keep on moving. So that has to be modeled perfectly. You should insert the particular loop perfectly. Proteination of your each amino acids are very important. Everything will take care when you prepare your protein. Once you do this process, the protein is now ready. Exactly. See here. What are the parameters that we are utilizing? Various data, optimization, and then using a charm, and finally you are getting your prepared protein exactly mimicking like your in vivo system. Okay, fine. I got my protein. Now, again the problem. Where is my binding site? Where I should define my binding site? Calculations. How do I define a binding site? If I know my cavity, absolutely that is good. If I don't know what I should do, how I identify? So don't worry because we are having a site search algorithm to perform that process. Often binding site will be curvatures, cavities, larger the site. Usually the ligand will go on both 1996 and 1998 itself. Lots of people have published the data considering and telling about a binding site. So by taking those words and thoughts, we are defining. So here also we have 3 different options. Receptor cavities. Once you define your ligand, defining your receptor cavities. That means you are telling that you don't know anything about your binding site. So automatically, site search algorithm, which is inclusive of your flat filling and eraser algorithm, will identify the receptor cavities. Okay, and then PDB site records, other hand. So PDB itself will say these are the active sites. So you can go with that site also active sites. And the other next one is the current selection. Say, for example, I know the certain interface residues to that I want to perform docking. Yes, of course, you can do by means of that. So we have 3 different options where you can perform. You can expand your site. You can down your site in case if you want to do global docking, expand and make the whole protein as a site. That in that way also you can do docking, global docking or blind docking. Either way you can do. In case if you want to perform these kinds of options, 4 ways. What are the ways you want to perform? Those options are available in Discovery Studio. Okay, that means of a bound ligand, cross linking, NMR studies, case bound ligand, anything you can. Now, I picked my binding site. Central assumptions very important. Good inhibitors must possess significant structural and chemical complementarity. Recently, some of my students whom I'm training, they asked me this question. They took some structure and then they looked into that binding site. And they told me that similar structure, there is a more complementarity. Why I am getting low box score? I asked them, have you analyzed your results? Yes, we analyzed your results. Are you getting hydrogen bonding infection? Yes, I'm getting it. How much the score? Still, the score is not perfect. Can you tell me what is the problem? I told them, have you checked the chemical complementarity? We were thinking, ma'am, anything a compound should go on mine, that is called docking, right? No, not like that. Always, some compounds, say for example, I'm taking some set of ligands, which is rich in hydrogen bonding interaction, hydrogen bonding acceptor, I could say that. And my binding site, even your protein is also a chemical molecule. It's not a biologically, it's a big chemical molecule. That's it. Your protein is also a chemical molecule made up of CH2OS. If that site is also rich in acceptor, though this molecule is going and binding shape complementarity is there, definitely will get low dock score because there is no complementarity is there. That's what very important. Considering shape and size, chemical concentration of charge and non-polar interactions are very important in your docking analysis. Even though shape complementarity is safe, if there is no proper chemical concentration, definitely you get a low dock score. Okay, so henceforth, when you are working on any docking tool, this has to be considered and have be given must consider the central assumption. If you want, you can read this paper, very excellent paper. You can get it from an internet, and he has explained very nicely in a science journal. Now, screening, high throughput screening. Okay, library of molecules. I have imagine 20 to 100 molecules. I cannot go for a flexible docking assets. So I'll be screening with a confirmation, cashless confirmation algorithm I'm using once again to generate a various confirmation sampling and choosing a specific confirmation using libdoc. In the libdoc, which uses a Lodi algorithm, always the positive highest score is considered the best. And CDocker, negative energy is considered the best. Flexible docking is a comparison of both algorithms. Based on the size, you have to do your screening process. So you can see here, high throughput screening in Discovery Studio. Prepare your ligand, prepare your protein, first screen with the libdoc. In case if you are having gold, you can use the interface in Discovery Studio. Optimize using CDocker, scoring using calculating binding energies. Here also you can use implicit Soren model, TBSA model, GBSA model can be included and to calculate your energy of your bindings. So CDocker uses the random replacement, basically uses the charm-based docking. Libdoc is a fast hotspot-based docking. Small molecular fragment, when you're doing fragment-based drug designing, we are using MCCS-based. And flexible docking is uses both libdoc and CDoc transformers. So for instance, I'm just pick the CDocker. So generating a high confirmation for your molecular dynamics, rigid random rotations. So CDocker is a grid-based molecular docking, employs a confirmation for your high temperature, simulating minimization, final output of your process. So you get a results like this, final process, and then see here we have a various options. That's what I say here. You want a diverse confirmations, you can check with the post clusters. You want a different with the river, you can check the simulation underlying higher temperature, cooling and dynamics, you can change this option. Orientation, different orientation with the Van der Waals threshold, you can change this parameter. But while changing, you don't need to worry about it. When you click automatically Discovery Studio help option will say you which option you are selecting and the respective citation. You can read the specific papers or the respective theory and you can understand before changing this option. That is very, very crucial. And then go ahead with your final process. And I get a results like this. You get a CDocker energy, higher the value indicates more favorable. Negative scores include an internal ligand strain and receptor ligand interaction. And interaction and negative scores indicates how much interactions are happening between your compounds. So finally, I sorted out my results. I will be checking for non-bonded interaction, analyzing ligand forces. Say, for example, I have a lakhs of forces. What should I do? That is also we are going to automate using ligand forces. And interaction filtration, specific interaction filtration also you can go ahead with your same ligand process itself. Analyzing all kinds of bonding interactions also can be done. This is how we'll be refining your results. So example of atomic level interactions. This is how the molecule and the receptor site will go and try to exchange your atoms. Once it exchanges atoms, again going back to the confirmation sample. And then we try to interact and form the bondings. And the maximum, the minimum the distance, that's how it matches. That should not have any spheric or unfavorable interaction. All the things will be taken and finally get the results. This is how you get the results. Distances are measured in terms of angstroms. And we can read and we can analyze various types of bonding interactions in Discovery Studio. If you are going to use visualizer, it is free. You can download from BIOVIA Discovery Studio Visualizer 2020-21 kind. And it can help you to understand both favorable interaction as well as unfavorable interaction. And the chemistry of interactions will be tabulated in your tab columns. Tab column will have a chemistry of interactions, distance, category, type of bonds, from substar, which atom to which atom it is bonded. All the options you can get it. You will get a publication quality results where you can change in different colors along with the 3D-dimensional and 2-dimensional interactions analysis also. An unless ligand process, say for example, you have a many number of more than 100 courses, it is not appreciable to do it by means of a manual intervention. That's why we automate the process to understand the table count, untable count, and then we are having a halogen count and other counts and statistical residue analysis will top your residues, which is involved in a tabled, untable hydrogen bond, which will be very, very useful. And along with the frequency of interactions also will be listed there. Now I sorted out 10 molecules. Okay, maybe I could say that 15 molecules are my hand. Still I am going for the process? No. Still I am not satisfied with the process. Next, I am funneling down into the molecular dynamics and C-energy calculation technique. Because always your molecules will be in motion, right? What we did a docking, even though a flexible docking, there are flexible residues are only given, not the whole molecules in motion. So to do that process, all the molecules are not static structures. Biological processes are mediated by movements and dynamics. Right from a simple vibration to produce a spectra, breathing of DNA, binding, understanding transitions, protein folding, all the things are happening in motions. How do I take up only the rigid crystals? That's why we are getting into the concept of principles of molecular dynamics. So can anyone guess what could be the motion we have studied from our school days onwards? This principle is for a motion, this principles of motion. Can anyone guess and put it in a chat? Maybe I am seeing, I cannot see chat right now. I'll be seeing only at the end. If you can think about it, just put it in a chat. Okay. And what could be the principles of molecular dynamics? Keep 10 seconds and counting. If you know what could be the dynamics, how we study the motion of a particle, think in terms of physical terms. Physically, how do we calculate? 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Fine. We see that who are posted. We definitely will connect you back for some gifts. Principles of molecular dynamics. So time, evolution, conformational space always will be their motion. And this will be governed by Newton's second law of motion. F equals to ma. Okay? So whatever dynamics that you are performing, it depends upon your molecular dynamics, Newton's second law of motion. Let's see at the end who will answer Newton's second law of motion or F equals to ma. Okay? So when you come to this process, there are certain vocabularies that we should know. Force field, energy minimization, ensembles, and then analysis. So these all are the 4 things that we keep on using. So what is a force field? Because when we are doing this analysis, there will be a missing atom to be there. But when we do protein preparation, almost 99% will be overcome. But if you're taking a raw protein, exporting from some other, you did a docking in some other tool, you want to export for dynamics, of course you can do that process. During that time, you are not prepared your protein, not follow that particular process. So in that way, there is a problem will arise. So in order to overcome the process, we are using a force field. Force field will allow to understand or display the missing atoms or residues that will be open and that will be updated with these specific parameters. Each and every position of an atom is very important and named in a different in a force field. See, you can see different positions of a same carbon as called different names. That's why we try to use a versatile force fields like a charm. Charm is a heterogeneous force field, can be applied for any system. And charm 36 also equally have a same kind of an atom types. In case if I don't have atom types, no issues. We are having that options also for you. Okay, water, ions, nucleic acid, ethers, and then second generation of charm, general force. All the parameters are present in charm 36. If you don't have atom types, no issues. We can edit the force fields here. So whenever you're opening or reading about a force field, you have what's a file called residue topological file. The residue topological file can be editable in this video. Because in case of carbohydrates or a lipid system, there usually this problem will arise in force field parameters. One can edit. How we can edit? So this is how the parameter file looks like, and residue files look like. So you have to copy and paste a particular parameter in the RTF file, and then you can copy and paste. And with that, so you can see how to edit and how to take from macro Maryland. Then you have a force field parameters for different RTF files that you can download, and you can copy and paste here, and you can import in this service. With that parameter, you can run, because non-standard amino acids have certain problems that you can rectify. And you can apply force field. When it's not typed, you can use that option. And we have a wide range of force fields in Discovery Studio. You can see here 19, 22, 27, 36, H, MMF, CFF, all kinds of force fields are available in Discovery Studio. So once you choose your force field, next is the energy minimization, because even when you are working with your modeling, your template model, homology model, we not get exactly 100%. It's not the same. Some of the parameters will be missing. In order to make your parameter or your molecule equivalent to your energy, your experimental technique, we'll be performing energy minimization with constraints and restraints. Also, we can do that. So it will help you to remove the back conformation, lax conformation, remove the steric overlaps and back context. So in the absence of your experimental structure, this will help you to minimize take your ideal geometry. For example, homology modeling application, homology modeling to improve the quality of the model, equivalent to your experimental model, we'll use the energy minimization. So we have lots of energy minimization protocols like the Stephen distance, conjugate gradient. Two things are very common. Apart from that, we are also having a Powell, smart minimizer, and ADMET methods. Any methods you can choose, but before studying or taking the tools, you have to learn about the parameters and algorithms. That will be very, very ideal when you want to understand in depth of biophysics concepts. And what are the proteins that should be solvated? It could be explicit solvated or implement solvated. So you can do both the kinds of solvent method, either explicit or implicit method. And in the implicit method, we have a centralized bond, molecular volume, and then polar surface area, and poison balls in non-polar surface area. Any different methods are available. You can choose any one of the methods. And this is the process. Take your protein ligand complexes, apply the force field. The protein ligand complexes are nothing but the one which we finished talking now. That is called protein ligand complex. Even for protein complexes also you can do this process. Apply the specific force field. Solvate the system. Either you can, if you're not going to solve it, you can use implement solvent method also. After that, use the minimization, heating, equilibration, production. Finally, analyze the trajectory. Check the deviation, fluctuation, and structure three-dimensional compatibility. This is the steps. Input your molecule, choose the algorithm. First algorithm, second algorithm. Why we are doing? Because sometimes if you're not minimizing your protein properly, that will create you errors. That's why we are trying to relax the conformations, and then we're going for heating, and then equilibration, production. And this is the integrated the shake constraint algorithm along with the integrator leapfrog vendor, which is again a mathematical formula. I don't want to integrate a mathematical terms. Just I'm telling that this is integrated with so and so. Very big algorithms are there, which is based on a Taylor's Francis, which we studied the Euler's rules. Whatever we studied in our standards, those things will be connected here, but we don't know application during that time. There is applications actually. So this is configured like that. Velocity terms and activity terms, first order derivative, second order derivative, lighter they derive. And then setting of a cutoff distances, higher cutoff distance and the lower cutoff distance for non-bond interactions. You can set. If you want, you can set, or you can leave as default. Okay. Other hand, people will, when you are doing a dynamics, suddenly your system is shut down, power cut off, electricity, like this, a problem will arise. One can restart your dynamics also. You can take up the restart file, which is created. From that, you can restart your system. And after doing a restart, you can process your systems. You can bring all your trajectory files and you can put together. You can restart, that is a trajectory file in DCG format. And finally, you can concatenate your results. After concatenation, you can study the RMSD deviation. This is analyzing tools. You can study the interaction energies, binding energies, all these things, and Delphi calculations using electrostatic also you can do. So, for example, you are doing for 10 different compounds analysis. You can select based on the least deviations between the compounds. When I see the compound 1, compound 2 is the less deviating compared with the compound 1. So I will go ahead with my compound 2. Similarly, fluctuation, your molecular docking process is a rigid. But when you do dynamics, you say whether your complex is keep on maintaining and the fluctuations are less between the atoms or not. You can study by means of root mean square fluctuations with respect to the secondary structure of protein. Next, various of variation will help you to understand your weighted RMSD of your molecule, whether your compactness of protein is maintained throughout the dynamics or not. Yes, of course, you can study that. So comparatively, I can see that compound 2 is constantly maintained. There is a decrease and increase is happening in the compound 1. So with that, when I started with the millions, I have ended up only with my 10 different compounds. Maybe I'm not sorted at finish because I am having free energy calculations. I can study hydrogen bonding interactions, various conformations versus my interaction analysis. You can see all these things are constantly maintaining its interactions. These amino acids are losing its interaction during a dynamics. And again, I'm not still ended. I'm studying interactions between my groups of atoms, whether my atom selection, residues of interface group between my receptor and then my ligand. And there will be terms will be added like this: interaction energy, van der Waals energy, and electrostatic energy. And then I'm seeing my interaction energy is negative or not. Yes, of course, it's perfect. Now, again, I'm not ended. Still, I'm doing, free energy, delta free energy, free binding energy calculation. Open the outputs of your confirmation and run it for your results for single binding energy trajectory analysis, free energy binding. So we take for complexes, your ligand, and then we calculate and give you delta G, final delta G for each and every confirmations. With that, I have only the more negative of delta free energy binding. Only those molecules I'm taking top 10 delta average delta energy molecule in expressing local mode. With that, I go for in vitro testing. Wherever we started, we came to the final conclusion. When I take my library, millions of molecules, I have ended up with my lakhs of molecules during this process, hundreds of molecules in this process, numericals in this process, numbers in my process, and finally I have only few numbers and I go for in vitro. This is a very, very cost-effective process, and you'll get a more reliable in your results. If you want to study initially, benchmark your process first. Take already processed, benchmark it for your ligands, then you start your study. That will help you and increase your research process and more reliable from your process. You never fail any steps. That's what I would say. You never fail any steps. You have failure, you have other options to perform your research. And you have an end-to-end solution in this service. So these are our BIOVIA government and academic partners, our customers in life sciences across the world, and Discovery Studio. So Discovery Studio, right now a macromolecule to small molecule optimization, DNA protein docking, DNA ligand docking, RNA ligand docking, all kinds of analysis can be done by using end-to-end software in Discovery Studio. So I hope it will be very helpful for everyone. With this, I will thank everyone for keep on constantly with me, clearing my session. Thank you all. Now, I think we can go for, there are lots of queries are there. We can start one by one. What are the queries you have? You can put it in a chat. Now I'm going to pick what are the questions or queries asked in the chats. Okay, coming down. Okay, Gurani Haita. Okay, explain about computational model. Could you little bit specific what computational model you want to analyze? Guranini Harita. Why removing next one? Anuva Mahesh. Why removing water molecule beta atom from protein, performing docking and checking a ligand interaction? Generally, no possibility to remove our body. Generally, what will happen if you are taking a molecule bound with your beta atoms won't be there in your systems. Water molecules will be there, but very rarely found in the active site. When your molecules are present, your water molecules are present at the active site, we should not remove. That's why when you click the protein report, that will say what are the crystal atoms or the water molecules near the active site, we should not remove that. Keep those water molecules and you have to run. If there is a metal in this particular question, we should not delete that metal also. So we should not do that. Something against a biological system we should not do. Mimicking like a system we have to do. Okay. When they do extracrystallography analysis, they will try to add certain atoms and they will try to add so many you can say carbohydrates also they will add to stabilize the structures. That has to be removed. Apart from that, everything we have to keep as is while preparing. And then Jagadish Chen, what is the difference between CDOC? LibDoc, so I explained it. LibDoc is conformational based and then Ludi algorithm. Higher the score, best the results. CDocker is basically a molecular dynamics based docking. CDocker will include even the ligand strings. When I say interaction, the CDocker interaction energy will, once the dock is finished, interaction will calculate. Interactions between a receptor and atom will be calculated. This is CDocker interaction energy. That's why we are having one more parameter called CDocker interaction energy. Because sometimes if people are very new to these kinds of net tools, they want to understand the binding energies. That's why we included one more parameter called CDocker interaction energy. Brownian movement. Brownian movement, any movement of a molecule from one position to another position, we use it using about Brownian movement. We are not dealing much. There is also the role is played, but we are not dealing with it. If you want, you can connect me back. I will send you how the Brownian movement is connected with the dynamics and simulations and also the kinetics. When you take a molecular dynamics and simulation, all the movements will be considered here. If there is a standard, no, I told you, right? There is no standard scoring metrics for ritual screening. If you have a pharmacophore based screening, higher the fit value is considered the best. Dr. S. Udayamani, right? Higher the fit score is considered the best. Next one. The reason is forming weird yellow rings around atoms. That is, say for example, that is to make a locations. Whether it will be moving here and there or not, it will show you around the atoms. Next, the difference between, so I think Jagadish just now I explained, right? Which is more validated research. See, more valid. First, you have to consider CDocker energy and take their corresponding CDocker interaction energy. That's how we have to take, because CDocker is having more role. It includes all the parameters. CDocker interaction energy is, as the name itself is saying, that it's interaction based energy. Okay. Anything else you want to? If you want to ask, say for it, ma'am. Yes, this session is in a live session in a YouTube. They already shared the link in the beginning. You can look up in that, and we post our email IDs, inquiry@altem.com. If you have any queries further, feel free to connect us. We'll be very happy to answer all your queries. If you want to raise your hand and you want to ask any queries, please raise your hand and then ask your query and unmute yourself. If you keep metal atoms and the important crystal water during docking, will they interfere during molecular dynamics? We have to, when we are keeping a metal atoms, and it will not, say for an instance, when you have your bound ligand or metal or water molecule, that will definitely will have an interaction. That interaction should not lose. Say for example, there are certain enzymes which will be activated only in the metals, which need to be retained as such. It will not. It will also plays a major role in a molecular dynamic simulation, which is accountable action. So that's what the water is also considered a pharmacophore. The metal which is bound with anything which is bound to active site is considered as crucial. Dr. Afsal sir. And
Yes, madam. Thank you, madam. So
they also provided feedback forms. So please click the feedback form. I think Google link form is provided, so that will help us for your further training and improvement purposes. And what is the next step after ligand? Next step after ligand and receptor spores. If you are there, Durani Harita, can you unmute yourself and then ask this one, because little bit I require the clarity. Please unmute
Hi,
yourself.
ma'am.
Hi.
Ma'am, my name is Harita. Hello, ma'am.
Yes, Harita, please.
Firstly, I would
Good
like
morning.
to thank you for the wonderful session. My question is to ask you about. I did my project in this docking, ma'am. I finished, like I selected a receptor and I took about 50 to 30 to 40 molecules, and I had this interaction free energy. I have this energy score, ma'am, for about
Okay.
3 or 4 compounds. The energy score was great for 3 to 4 compounds, ma'am. Now,
Okay.
what is the next step after that, ma'am?
Okay. Now we have your compound and you're sorted based on the energy values. Next step, you can take it for a molecular dynamics and simulation. Study how in a flexible motion or in a motion whether the interactions are keep on maintaining or it will tend to lose when we apply any external pressure or temperature. Study that. After that, include your, if case, if possible, include your MMPBSA method, molecular mechanics based Poisson-Boltzmann method, the non-polar surface area method, MMPBSA or GBSA method. It would be very, very ideal when you go for an in vitro testing. In case if you don't have, okay, no issues. What are the molecules you are taking? When you are following a good pattern in your molecular dynamics and simulation and constantly making its interactions in hydration bonding, then you can pick the molecules and further you can go for your in vitro test. Okay.
Ma'am, can you explain it once again, ma'am? I didn't get it properly. You said molecular dynamics. After that?
Yes. You do molecular dynamics and MMPBSA method.
MM?
TBSA.
Oh, okay.
Okay.
After that?
After that, go for in vitro test.
Okay, ma'am, one last question, ma'am.
Yes,
Whatever I have the best score for about 3 to 4 compounds. That's it, ma'am. Shall I do molecular dynamics for all of the compounds or the
Yes.
molecules with the best scores?
Best one also you can choose because you already started out based on the 4, right? Because molecular dynamics simulation is a little bit computational cost-based technique. Okay, you need a very good system to perform that process. So instead, you can take your prioritize based on your energy values, right? Take only 4 compounds that you prioritize and go for this molecular dynamics simulation.
Ma'am, one last doubt, ma'am. One last doubt.
Please. Yes, please.
Actually, you said something about the bending and steric overlap. I made a molecule according to ChemDraw, ma'am, I made a molecule in a ChemDraw structure.
Okay.
I didn't see that steric overlap and all, ma'am. Will it affect the ligand score, ma'am?
See, when you're doing a docking, have you prepared your ligands?
Yes, ma'am. I interacted the ligand with the molecule, ma'am. But I didn't check about that steric overlap and difference in the bond angle. I didn't check that properly, ma'am. That parameter properly. Will it
Okay.
affect that ligand score?
Definitely, there will be a say. For example, I will ask you just answer me yes or no. When you draw your structure, have you seen the geometry?
No, ma'am. I haven't seen the geometry.
Okay, that means sketching is different, optimizing is different. Everyone can do sketching. Say, I can pick the molecule, I can draw it. That doesn't mean my carbon-carbon atoms are perfectly done. There will be
Oh.
a distance because for carbon to carbon, carbon to nitrogen, carbon to oxygen, there is a certain distance out there. If you don't do clean geometry, okay, that will affect definitely your interactions also.
Oh.
So whenever it's not, yes, please, yes, please.
Yeah, ma'am.
Yes, Harita. See, you are doing with a chem sketch or chem draw. Whenever you're drawing, there is an option called clean geometry in any tools. First,
Okay.
clean your geometry and then import your ligands. Even Discovery Studio Visualizer is also available for free. If you don't have this option also, there is available. You can open that ligand where you can optimize it. Open and optimize, and then use export your ligand wherever you want to do docking. Save it in SDLCF file and then go for docking. That will correct your problems. Okay.
Okay. Thank you so much, ma'am. Thank you.
Okay. Welcome, Marika.
Thank you, ma'am.
So hope there are no other queries from the participant side. So Madam has given clear explanation about each and every module in the Discovery Studio. So thank you, Madam. Thank you for your thought-provoking session, and it's very informative about all the tools which are present in Discovery Studio.
Yeah,
Thank you once again.
Benefit from our
Yes, Madam. I think most of the students from different various colleges, they mostly represented here in this webinar. And finally, I welcome Dr. Gauramma Madam for vote of thanks.
Thank you, Srikanth. Good afternoon, everyone. As we come to the closing session of the live webinar on state-of-art molecular modeling tools, BIOVIA drug discovery suite. It is my pleasure to deliver the vote of thanks to all who have helped to make this event such a way. I take this opportunity to thank our Pro-Chancellor, Dr. B. Suresh, for his continuous support. On behalf of our management, JSS Academy of Higher Education and Research, Mysuru, and JSS College of Pharmacy, Ooty, we would like to express our deepfelt thanks to today's speaker, Mrs. Divya Sanmugarajan, Senior Application Scientist, Life Science, Atlam Technology Private Limited, Chennai, for sharing their knowledge, precious time with their interactive session to all participants. We thank the delegates who came from different places have joined us at the event. We extend our heartfelt thanks to our beloved principal, Dr. S.P. Dhanabal, for being with us and inspiring us. We would like to express our deepfelt thanks to Dr. Muhammad Afzal Azam, Professor and HOD, Department of Pharmaceutical Chemistry, and Dr. Komadi Swaminathan and Dr. Srikanth is a coordinator of today's program, and Mr. C. Jaykumar for making this event a great success. I would like to express my heartfelt thanks to faculty of Department of Pharmaceutical Chemistry, all staff members, research scholars, and PG students of Department of Pharmaceutical Chemistry for making this event a great success. I thank all staff members and students of this institution for their kind cooperation to organize this function in such a good, grand manner. Once again, I thank each and every individual. Thank you, one and all.
Thank you, Madam.
Thank
Okay,
you,
thank
sir.
you.
So with this, I will also express my great gratitude to the JSS College of Pharmacy and their staffs because I know about JSS. I have been keep in touch for more than 6 years with JSS, Mysuru, now into UT. So with this, I'll express my gratitude to Pro-Chancellor Dr. V. Suresh sir, and then Principal Ganapal sir, and then HOD Dr. Afzal sir, and then the respective staffs and the teachers and the attendees and delegates for everyone for attending the session and make this on a great success. Hope this will lead your seed in you all and make you great changes in your research.